Okay, well, I think we'll get started. Thank you all very much for coming, and I hope everyone's having a nice weekend. So I'm Ted Huey. I'm the relatively recent new director of the Memory and Aging program at Butler Hospital. And I'm going to speak a little bit on Alzheimer's disease, some of the work we're doing, some on new treatments, which have been in the news a lot. But actually, I want to reserve the bulk of the time to have two very, very talented one Brown alumni and one current Brown student speak about their experiences getting involved with research. So this is Natalie, Riera, and Joyce here. Because I think that it might be very interesting for people to hear about kind of what their experience has been, how they got involved in research as undergraduates, and in Hannah's case, who's graduated, kind of what that's led to afterwards, and then kind of tips for people who might want to get involved in research as an undergraduate. And I have yet to meet an untalented Brown student, but they are both particularly talented among Brown students. It's a pretty impressive crop of people. I'm always nice to them because I'm never sure who I might be working for in the future. So, okay. This is, not that big of a group. So, we'll have some time for questions at the end, but also we can stop in the middle and if you see something you want to ask about. To get money from NIH. So I'm going to talk a little bit about some recent developments, some specific to what we're doing here at Brown and at Butler Hospital, but then also some things that have been going on in the news. Those of us who work in this field, and I've worked in this field a while, feel like sort of, things were very, very slow and nothing much changed for like 15 years. Now it's like every week I feel like something new is coming out and you have to, keep an eye on the newspaper. So we'll talk about some of the new things. The big one being the first new treatments approved for Alzheimer's disease in about a decade or so, which just happened in the last couple months. And then also this interesting thing, which also has just been approved, but of what we call biomarkers, which are biological tests for Alzheimer's disease based on blood. Okay, these are kind of stuff that you probably know already, but the kind of obligatory slides showing what an enormous problem Alzheimer's disease and related dementias are. And it is an enormous problem. I mean, we're in the midst of, I consider it an epidemic, and I think people usually think of epidemics as being infectious. This is not an infectious epidemic, but really as our population ages and the world population ages, the number and proportion of people with Alzheimer's disease increases dramatically. So this is projections starting at year 2020, but then projecting on given the rates of growth of the population. And you can see just going up and up and up, especially in the oldest old people, ages 85 and older, but even expanding in younger people as well, to the point where coming from about 6 million now, but going up to almost 14 million people in the United States. The other depressing slide, which is that we really have not done as well as other fields at getting on top of this. So what this slide is showing you the change in how the proportion of overall deaths in the United States due to different illnesses. And so you can see here we start with cancer, which has remained pretty stable, maybe gone down a little bit. Heart disease, this is starting I think in, when was this, 2020 to 2019. Heart disease, which really had its main benefits actually before 2000. So this lowered quite a bit before 2000. HIV, a lot's come out. Now HIV is not a fatal diagnosis anymore. And so that really has gone down quite a bit. The proportion due to Alzheimer's disease has gone up. And not that we've gotten worse at treating it. It's just that as we get better at treating these other things, the proportion of deaths due to Alzheimer's disease in the country go up. And this reflects the fact that we really have not had any disease-modifying treatments for Alzheimer's disease until recently. This does not affect the country equally, so we know that that non-white racial and ethnic groups are particularly affected. In the Hispanic and Latino communities and African American communities, the prevalence of dementia is significantly higher. And we're still kind of working out why that is. It's almost certainly multifactorial with some genetic aspects, some social determinants of health, and others, but it is more prevalent and more of an impact in non-white communities in the United States. This is a very kind of rough model of what we think happens. You guys have probably all heard of amyloid protein. Is that so? There are two proteins which are the pathological hallmarks of Alzheimer's disease. One is amyloid, which forms plaques, which are collections, extracellular collections of amyloid protein. And then there's another one called tau, which forms tangles, which are intracellular clumping of tau. And so sometimes you'll hear about plaques and tangles, and that's all it is collections of amyloid, collections of tau. We think that amyloid accumulation, this doesn't have tau on there here too, but also tau accumulation, starts before people get symptomatic. So that now that we're having better and better biomarkers, we can find a significant number of people. If I go out onto the street and pull out a bunch of elderly people, some will be positive on amyloid tests, indicating that they have significant amyloid deposition. They may be completely cognitively normal, but what it means is that they're at risk for developing dementia and Alzheimer's disease later. This term we use, so a little bit of the terms which can be a little confusing. This term MCI stands for mild cognitive impairment, and the idea behind that is that nobody wakes up one day and has dementia due to Alzheimer's disease. Everyone passes gradually through a phase where they may have some memory problems that make it harder to get by before getting a full diagnosis of Alzheimer's disease or dementia. Another terminology thing that's confusing is what's the difference between Alzheimer's disease and dementia? Dementia is just a clinical diagnosis and term, and it means that you're having cognitive problems to the point where you can't live your daily life in the way you used to. There are many causes of dementia. Alzheimer's disease is the most common, but there are many other causes. So, with the terminology, dementia is the clinical state people are in, that they pass through mild cognitive impairment into dementia, and the most common cause of dementia is Alzheimer's disease. Any questions or thoughts at this point? So here at Brown, we'll talk mostly about kind of national things, but just to highlight one project. I mentioned before that the distribution of dementia and the distribution of the particular symptoms within dementia are not uniform across different groups within the country. And we have, as a field, been working quite hard to enroll enough research participants of different ethnic and racial backgrounds to be able to really kind of study this problem and study this issue. And we're very excited at Brown. Just about a month ago, we got a U Award, which is NIH speak for sort of the contract from the government to examine neuropsychiatric and other non-cognitive symptoms in different ethnic groups. And you can see we finally kind of gotten to the point where we're having some numbers here where we can start to do genetic association studies to determine some of the genetic causes of some of the symptoms of Alzheimer's disease. We have over 80,000. By the time the study's done, we're going to have well over 100,000 and quite diverse. And these pictures here are just showing that when we kind of have people self-report what their ethnicity is, it's very gross. And really what we want to do and what we do with the genetic studies is we do what we call ancestry mapping, which is we go through the entire genome, and all of these 100,000 people are going to be entirely sequenced. So every base pair in their entire genome. And what we can do is kind of assign different loci in different parts of the chromosomes from different ancestral backgrounds. So here is someone who is African American, and red are genes of African origin. And then here is someone who is non-Hispanic white, where blue is European origin and red is African origin. And here's someone who's Hispanic, where blue is European origin and yellow is Native American, and red is African American. And you can see, especially in someone who is self-identifies as Hispanic, it's not simple. And so if you're trying to understand the genetics of Alzheimer's disease, you have to do this kind of fine ancestry mapping. And of particular interest we've highlighted in this is that ApoE, which is a gene that's associated with Alzheimer's disease, is actually in this person of African origin, even though they are Hispanic. So we're excited to be starting this study. It's just like it just got off the ground. Okay, so now moving to more kind of general stuff. Biomarkers, so again, this has been in the news a lot. We had for a long time what we would call cerebrospinal fluid biomarkers, which was where we took fluid out of someone's back, and we would actually, some of it's not rocket science. We would send it for amyloid and tau protein and look at the levels. And from those, we knew the pattern of tau and amyloid protein that were suggestive of Alzheimer's disease in the brain. That we've had that for a long time. More recently, we developed something called PET scanning for specific to amyloid. And so what that is where we inject someone with a mildly radioactive molecule. It doesn't expose them to a lot of radiation, but it's enough that we can pick it up, that sticks to amyloid specifically. So it goes into their body, we just inject it in a vein, and then we put them into imaging. And this is what we see. This is kind of color mapped here. And these are 3 cases of people where they died shortly after having the scan. And so here's what their scan looked like. And then when we did an autopsy and looked at their brain, this is what their brain looked like. And this kind of clumpy stuff, these are plaques. These are amyloid plaques. And so you can see a pretty tight association between this person really doesn't show up much on the PET scan, and they really don't have any amyloid plaques in their brain. This person has moderate increased color. So the hotter the color, the more binding there is. And you can see they've got moderate plaque burden. This person has really pretty severe plaque burden, and you can then see it in the scan. So these scans are really quite good at telling us how much amyloid does someone have in their brain. And of course, the advantage, we can do it while someone is living. That's where we were for the last several years. Just in the last year or two, blood-based biomarkers have taken off. So what these are is they're like the PET scans, they're like the cerebrospinal fluid, but we can do it on blood, which is obviously a lot easier to do than a PET scan or a lumbar puncture, which is the procedure we do to get cerebrospinal fluid. So, and this is a busy slide. There are lots of different things, but this is comparing to PET scan and this is comparing to cerebrospinal fluid. The point just being that these are people, so and this axis is people who have amyloid on their PET scan, and then this is what their blood-based biomarkers show, and showing kind of increased amyloid, and the same thing compared to CSF. So now, just as of a couple months ago, these are now clinically approved. So I can write an order, and someone can get a blood test, which tells you how much amyloid essentially is in your brain. Now, one thing to keep in mind with these is this isn't a test for if someone is sick. So like I said, if you kind of can go out there and you can find people who have normal cognition or living their lives but may be positive on these biomarkers, we're still kind of not sure what that means. Probably that person is at much higher risk for developing dementia in the future, but we're not sure when or how that will be or what that will look like. And so some people say, well, I want to get this and it'll tell me kind of, what's going to happen. It's a little more complicated than that. For people who are symptomatic, who have either mild cognitive impairment or dementia, and then we get this test, we're pretty sure at that point we know why they have mild cognitive impairment or dementia. For someone who's cognitively normal and gets this test, we're a little unsure how to interpret that. So at the moment, we don't recommend people who have no symptoms getting this test, just because it's very hard to recommend. Yeah.
What is the difference in sensitivity between blood and CSF?
So it used to not be good. So that's why we relied on CSF for a long time. And we, again, for decades we've been sending CSF. It's gotten quite good. And that's actually this graph right here. This is CSF. So what the amyloid is on the CSF versus what, it gets a little confusing. I don't want to get too much into the weeds. We actually look at amyloid by looking at tau in the blood. It would take a long time to talk about, but it's quite good. So the association now is good enough where we feel like we can start to use it in the blood.
So essentially you're saying we don't need CSF anymore.
We don't need CSF. Let me qualify a little bit. This was clinically approved, the blood tests 2 months ago or something. We're still really it is Procivity. They have a panel now, but other people are going to have panels as well.
Is it reimbursed with CMS?
Not at the moment. It's pretty expensive. But one would imagine, A, it'll maybe be reimbursed by CMS, or B, the cost will come down. This is, actually just today, this morning, or I guess it was yesterday morning, sorry, it was Friday, CMS approved PET scans. So those are now paid for, which is a big jump. Sure.
What percentage of people that have AD do not show this biomarker?
Yeah, well, that's a good question, and it's a great question, and a little complicated because, it depends a little bit on who you ask. One possible answer to that question is, well, if you have dementia and you're negative on these biomarkers, they're good enough that it's probably not Alzheimer's disease. Now, that's not 100%, but that's pretty good. So there are how I said Alzheimer's is the most common cause of dementia? It's probably one of the other causes at that point. And they're good enough that you can probably say that with pretty good confidence.
So this is 100%. They think there's that.
Not 100%, but it's pretty close to the point that, so I study, actually, I'm particularly interested in non-Alzheimer dementias. And we use these tests often. So if someone has, I was just talking to someone about frontotemporal dementia, which is another type of dementia. If we have someone coming in and we think, well, we're not sure if they have frontotemporal dementia or Alzheimer's disease, we'll get these tests. And we'll say, well, if it's positive, it's almost certainly Alzheimer's disease. If it's negative, it's almost certainly FTD, even though we don't have a test for FTD. So probably if it's negative in someone who has a dementia, has a full dementia syndrome, you should look elsewhere of what's the cause.
Yeah. So what are like the positive implications of this like early identification? Like I've read about how like with amyloid specifically, like these patients would know that they're at risk for it, but like once they have that information, there's like nothing they can do with it in terms of like insurance system.
Yeah. No, it's a great question. The one big reason, if I can get to the treatment section, because that's changed a little bit. I'll give you the short answer now, which is that all of the new treatments are only tested and approved in very early illness. So only at the MCI or very earliest dementia stage. The implication of that is you don't want to wait too long to pick it up and to identify it. And all of the treatments demand now a biomarker. So really kind of they've become, and that's why CMS, which is the government agency that approves whether the government will pay for testing, approved amyloid scans yesterday. They're pretty expensive. They can be up to like $10,000. They're expensive. But the reason that all has moved forward and with the blood-based biomarkers has been pushed a lot by the treatment because people want to get treatment and want to get into treatment early. So that's been the main thing that's changed. But it's a really good question and good point. I'll move kind of quickly. I want to be sure our students and former students have time. But into the treatments, we have now one approved treatment, lecanemab, which is an anti-amyloid antibody. So basically, antibody is the way your body immune system clears things from your body. And basically, what we do is inject people with an antibody directed towards amyloid. And we know, and this is data, you're not going to get clearer data than this, that it clears amyloid. So this is higher doses as you go down of lecanemab, the agent we have approved now. And then when you do those PET scans and say, afterwards, how much amyloid is left in their brain, you can see it goes down, down, down the more you give. We've had these for a while, these kinds of amyloid-lowering treatments, and have not yet had one that really had a clinical impact until lecanemab. It's also called Leqembi. And then this graph here, may seem silly, but this is like a multi-million-dollar graph, and that this was the data they got approval on. This is basically saying, if they had the 18-month trial, if you were on the anti-amyloid antibody for that time versus on the placebo, you declined less quickly. You didn't get better. It didn't cure it. It didn't take it away, but you don't decline as quickly. The scale here is something called the sum of boxes of the CDR. It's an 18-point scale, and it's half a point on there. So, I think it's very important to kind of flag, how clinically important is this? It was enough to get approval. It is significant. It's not a cure. And I think that, be skeptical of people who say, oh, this will, clear it and then you're fine. It's not. It is an advance. We're going to start using this in our center, but I think we should all kind of have open eyes about what it is and what it isn't. Questions or thoughts on that, or people heard?
I kind of some controversy around this. So there's a bunch of controversy.
Yeah, some of the controversy was historical. So there was one medication called aducanumab or Aduhelm, which preceded lecanemab, and that got approved, and the data wasn't nearly as strong. Lecanemab and donanemab, which is the other agent, the data is a lot stronger. I mean, I think most of us, myself included, and I've gone over these data quite carefully. This is a real effect. The question is how clinically significant it is, but it is. This is a real effect we're seeing. The aducanumab, it was much more questionable, and so a lot of people say, well, that shouldn't have been approved. Lecanemab and donanemab is not yet approved, but I don't think anyone's really kind of said, oh, they shouldn't be approved.
The one thing I want to ask you is, hasn't been really established that if you remove the number of plaques, that results in better cognition? Is that being clearly established?
So, you're asking great questions. Again, complicated. So this is not the first medication to reduce amyloid plaque. We've had those for a while. And whenever we've done clinical testing, they haven't been helpful. Now, this is a somewhat different, you can direct the antibody to kind of different parts of the amyloid, and this has a rationale that it's somewhat different from the other ones, but it has to be, I think you have to account for this previous literature of it not helping. And everyone was kind of puzzled by that. If amyloid is a big part of Alzheimer's disease and you clear it, why aren't people better? The point, I think, still holds. I showed you that graph here. You're really clearing amyloid. Why are they only half a point better on an 18-point scale? You would hope or think that, people would kind of like, come right back up or, but they're not. So even though we now have the proof, I think your point, the question you're asking is still very valid and very important question. Why is it not more? And I don't really have an answer for that. Yeah, go ahead.
I've also read that like in the past 30 years or so, all the funding has just been, all the research has been focused on amyloid. How strong is the connection between Alzheimer's and amyloid to the point where are there any other pathways besides tau that are being considered right now?
Being considered, but not nearly as well worked on. So tau, tau correlates better with cognitive symptoms. If you do a tau scan and then how demented someone is, it does better than amyloid. There are also these people out there, and I've been referring to them, but you can have people who, when we look at their brains, they've got a ton of amyloid and they're not demented. There are these other pathologies. So I'm particularly interested in one called TDP-43 and other pathologies which appear to be kind of adding into the mix. The model I think we're moving towards, and this is kind of really pushing the envelope, is more of an additive pathology model where if you have one type of pathology like amyloid or tau, it increases your chances of being demented. If you have 3, it's much more. 4, it's much more. 5, so it becomes more, I think a model again might be sort of similar to cardiovascular disease where if you have diabetes, your risk of having cardiovascular disease is increased. If you have high blood pressure and diabetes, if you have, I think we're kind of moving towards that model. I'll skip the side effects, but it is something also to think about. There's this one side effect, I'll just say quickly, called ARIA, which is probably the thing we worry the most about. Which is where people get leaking either of fluid or blood into the brain. And some of that we think as we clear the amyloid is kind of really wrapped around a lot of the vessels and they can get leaky. And there's actually some early data that those who have more of the, especially the asymptomatic, so they're not symptomatic, actually can have a bigger therapeutic effect too. That it is part of the kind of, what we're seeing with ARIA, which this leakage may reflect how well we're clearing the amyloid. But it is something we have to watch for. And we watch for it by doing lots of MRI scans. So this is the current recommendation where we do one before. We also do a PET scan that I mentioned, and then we do several afterwards. Something just to note, and I won't spend a lot of time on it, but this is very, very expensive. The drug itself is expensive. The scan is expensive. What do we got here? 5 MRIs, it's expensive. And one issue that we're concerned about, and I don't have an answer to, but is that this doesn't become something kind of that is not offered to people without money, which I think we're in danger of, I would argue. We just want to note that non-pharmacologic treatments are also, we have a trial going on at Butler for this, and there's a lot of this, we're finding out more and more that risk factors that affect cognitive impairment are modifiable, especially lifestyle factors, exercise being the big one. But also cognitive stimulation, general health improvement, nutrition. And we have a trial where we are intervening with all of these in people who are cognitively normal and seeing how much we can reduce the incidence of dementia. And these are side effect free, inexpensive, we recommend to everyone. So, and there's significant, up to 35% may be reducible by these risk factors. And when this literature first started coming out, I was skeptical. I was like, I don't really believe it. It's been maintained really over the years. So probably where we're going with the drug treatment is, and you brought this up, probably to combine treatment. So if you look at HIV, no one gets one HIV medication. They hit HIV kind of at multiple places. Probably what we're going to do is you'll come in and you'll get your amyloid scan, your tau scan. Basically we'll characterize someone's pathology and then give agents targeted against them, and then hopefully get more of an effect. So I'll stop there unless anyone has any questions. Yeah.
I wanted to ask you about nutritional counseling. What do we know for sure about dietary changes actually reducing the risk?
Both the dietary, it's all part of a package, and I feel it's sort of funny because I'm interested in dementia. I now feel like I'm a cardiologist because basically all my patients, it's cardiovascular health. So low saturated fats, all the stuff that's heart healthy, and a lot of this is we're recognizing scientifically very strong connections between vascular health and these pathologies. That if your vessels aren't feeding your brain well, you're much more likely to develop these.
I've also been reading about sugar as being pro-inflammatory in the brain.
Yeah, I'm not sure. Sugar, I don't know about that literature specifically. There definitely is developing an inflammatory literature, which could be linked to the vascular. But yeah, so, I had on my little theoretical future list, probably will be targeting inflammation as well, too.
The neuroprotective drug, what is that?
Well, we don't have those at the moment, but there's lots of interest in this. So kind of taking other approaches of things that may increase energetics, other things that are kind of helpful for neurons. But this is all future. This is not. There's nothing I can list here at the moment. Yeah, it's okay.
I remember reading about cognitive reserve.
Yeah.
And how like you could prevent it by exercising and word puzzles, but then once you start getting symptoms, it accelerates. Are you guys?
That's been supported. So before I came to Brown, I was at Columbia for 13 years, and the big cognitive reserve guy, Jakob Stern, is one of my mentors there. And it's a fascinating finding. You said it very well, but basically, just to repeat it, this idea that if people have, a lot of mental stimulation, things like education, things like that, they're actually protected against the pathology, so that they do better on the testing, even with worse pathology. But then one of the kind of biological implications from that is they kind of can fall off a cliff because they can compensate, and then they just can't do it anymore. But that is supported clinically. Yeah.
What's the connection between gut microbiome and inflammation?
That's really, so I just went to an NIH when I was going for the genetics. They're putting a lot of money into it. I don't know enough to speak, but there's some really interesting people working on that. But it's a topic that is you guys are really up on all the stuff. You're pushing me beyond what I know. But it's a topic that is really of great interest. They're funding a fair amount of research in that area. Okay.
What's the latest on we're talking about cardiovascular health, statins and dementia?
Yeah, that's so overall, there were some reports of statins being associated with cognitive impairment themselves. And so it was relatively rare though. And so a lot of my patients were coming and saying, oh, should I get off my statin? We have generally recommended people stay on it. And the data suggests it's probably worse to have high cholesterol, go off it and have high cholesterol, even for cognition. We kind of take it on a case by case basis, but as a population, we've generally recommended people stay on their statins.
Is Alzheimer in any way connected to autoimmune, or is that connection being researched?
Well, this inflammatory side, we're really, there's something there. And we've tried actually, I'm old enough to remember we tried anti-inflammation trials. We tried ibuprofen, we tried steroids even, things like that. We didn't get really traction on them, but it's kind of we're getting better at understanding it. So that's kind of something that's really going to develop, I think. All right. Okay, one last thing. I want to be sure there's.
In what way is statin causing the problem with the
Yeah, that's a great question. I don't actually know the mechanism of it. I know it was a rare association seen. I don't know the mechanism. That's a great question.
usually everybody swears by the statin.
And it's very rare. I mean, one thing I stress to my patients is it's better to have your cholesterol under control than to worry about the cognitive effects of statins, because it's uncommon. It's quite uncommon. All right, I want to leave enough time for this. Thank you, guys. Great questions. You guys don't need me to talk. You guys already know all this stuff.
Thank you so much for that, Ted. Can you all hear me good? Okay, great. So I'm going to shift gears a little bit. My name is Natalie. I'm a current senior here at Brown University studying neuroscience on the pre-medical track. I'm just going to talk a little bit about my experience getting involved in research. I've been conducting research for about 4 or 5 years now, so I feel like I'm pretty well versed at the moment. I know a lot of students here are probably interested in getting involved to some extent, or you have kids that are maybe interested, so hopefully I can be helpful. So like I said, I study neuroscience. I'm from Miami, Florida, and I first became interested in Alzheimer's disease when I was around 13 years old, and my grandmother was diagnosed and then later passed away right before I came to college. So that really just piqued my interest right before I came. And I actually started working at the Memory and Aging program with Dr. Louisa Thompson my freshman year. And I've worked on 3 projects with her. I've received 3 UTRA awards and 1 Link Award through her. And I've also worked at Columbia University this past summer with Ted's colleague at the STAR U program. So that was an NIA MSTEM funded grant with the Advancing Diversity in Aging and Research through Undergraduate Education. So those have been great opportunities, and I feel like Brown has really facilitated these opportunities as well as the Memory and Aging program. So like I said, I worked with Dr. Thompson on 3 different projects. And this past summer, I did some wet lab research with Dr. Ramachandran at the Ramachandran lab there. So I have some experience in both clinical research and wet lab research. So I feel like I can weigh in on the pros and cons of both of those. So with clinical research, I really enjoy more patient contact that I've been able to establish there. So my plans after college are to pursue medical school. So I really enjoy having that one-on-one patient interaction that working at the Memory and Aging program has really allowed me to do. You are also more in control of your hours because it's kind of like a 9 to 5 when patients are allowed to come in. However, you do a lot more data analysis and sitting at a computer, which may not be kind of the fan favorite of a lot of students, versus wet lab research is the more traditional field of research that you think of when you think of someone studying Alzheimer's disease. You see them in a lab with a white coat on. So that involves animal or cellular research. I did in vitro and in vivo this past summer. So we had mice in the lab, which was exciting. That also, however, means you have to wait for all of your experiments to process. So it kind of depends on if you would rather work with patients or work in a lab. There are benefits to both of them, and I've found both of them to be very, very rewarding. I think I've decided I prefer clinical a little bit more, but it was still a very worthwhile experience. And I probably would not have done anything different. So research here has definitely opened a lot of different doors for me. Like, for example, this past summer, I wouldn't have had this opportunity if I had not done the research here. And just being a member of a lab really facilitates your career post-grad. It allows you to work in this kind of more fast-paced adult environment, and just building rapport and relationships and mentorships that would definitely last a lifetime have been very important to me, at least. And everyone has been very helpful in my journey through college and to medical school soon. I'm so sorry. Okay. So my next steps include, I'm currently studying for the MCAT to take it in January and apply this upcoming summer. So I'm planning to take a gap year before medical school so I can apply during gap year. And I probably will work as a research assistant somewhere. Still figuring that out, but that's the plan. And hopefully apply to medical school and attend in 2025. So that'll be great. I also wanted to talk a little bit about the specific research I've conducted and the different studies I've worked on. So at the Memory and Aging program at Butler with Louisa, I have worked on the DigiCog study with her. That was my first study that I started freshman year. Like Ted mentioned, there's not a lot that could be done with Alzheimer's disease. So I was particularly interested in better diagnosing practices. So currently what we have are the PET scans and biomarker approaches, but also our cognitive assessments are a little bit outdated and not very sensitive. So I was interested in applying those to better encompass a wider range of the population. So I worked on the DigiCog study my freshman year, which has since concluded, but that study basically encompassed trying to digitize these cognitive assessments in a better manner. So we sent people home with kind of iPhones or smartphones, and every day they were prompted to do these assessments and mental games kind of, which would then come back to us, and then we would compare those as well to their cognitive assessments, which could be very promising in the future, especially with advancing technologies and just reusing the same cognitive assessments isn't very helpful in the long term, especially if it's not sensitive to differentiate between things like MCI or more progressive dementia until it might be too late. I then worked on a sleep study with her, which was part of our greater scheme RES 2.0 study. So there is a lot of speculation that poor sleep can be an early indication of early Alzheimer's disease. So we sent patients home with watches to wear for 2 weeks that would track their sleep data and then compare those with their cognitive assessments and their biomarker and PET scans. So that was very interesting to see as well. And we have a manuscript that came out a few months ago through that. And right now we're working on the RES 2.0 study. So that's looking at the Atlas retinal imaging of Alzheimer's disease study. So we are looking at a retinal imaging process as a new biomarker. So like Ted mentioned, right now the biomarkers that we currently have are cerebrospinal fluid biomarkers, which include a spinal tap, and it's not the most comfortable thing, a little bit invasive. And also PET scans are very expensive. Not a lot of clinics have a PET scanning machine. So it's not very accessible to many patients, very few patients actually. So we are interested in looking at more accessible approaches to these biomarkers. And the plasma biomarkers seem to be hopeful. We're also looking at retinal imaging as a possible one. So we have this kind of eye scanning machine that you can normally use at the eye doctor. So it could be very applicable to different clinics. And the eye has very similar cells. So your retinal ganglion cells are very similar to neurons in your brain. So you can see early indications of these amyloid and tau neurofibrillary tangles through this, which could be very promising. So that's what we're currently working on now. And this past summer at the STAR-U program that I did, I specifically looked at neuroproteosomes and how aberrant proteins are affecting neurons and how they recognize and respond to those. So specifically, how do neurons respond to amyloid beta, tau fibers, and TDP43? So there is a specific pathway that we looked at through this neuroproteasome leading these neurons to apoptosis or spontaneous cell death, which could be very promising as well if we look further into the RNA sequencing that has to do, and kind of see with these new medications that are coming out, are we not seeing that many results because of this neuronal death is a main thing I'm interested in, because once these proteins kind of attack the neurons and you lose the neurons are non-regenerative and you can't regain this lost function already. So that's some of the research that I have been working on. I'm interested to see where this takes me further, but we're going to be wrapping up the ARIA study, hopefully around May, getting some results with that. So that should be very interesting. But yeah, that's a little bit about my experience here at Brown and the research I have done. Does anyone have any questions at all? Yes.
It sounds like your research has helped you find internships, may help you find a job, maybe now. Application or that. How did it help you, if at all, with your schoolwork?
So it's definitely led me down a rabbit hole of specifically Alzheimer's disease. I study neuroscience, so in all my neuro classes, I feel like it's definitely helped me understand at least the more molecular level kind of approaches in some of the classes, just because Alzheimer's disease is so convoluted. I feel like it's definitely all positive experience. Yes.
As a physician, I want to say how excited I am that you listened to join us. You just did a fantastic job. Well done.
Thank you so much.
And I'm interested to hear, having worked a little bit with TDP43, this may be a very unfair question, so feel free to say no, not my thing. But do you know if the field is evolving toward measuring new biomarkers of TDP43 protein?
I can definitely see that evolving into something. The study I participated in this past summer was like the very, very early stages of the study. I was working alongside a PhD student, and this was her dissertation study. So hopefully, yes, I do see some promising things there, especially when we looked at the neuroproteasome and its interactions there. I specifically looked at the PERK pathway, if you're familiar with that. There was definitely some interaction there with leading the cell to apoptosis. So I would say yes, but.
In your neuroscience classes and in research, have you seen any correlation between people who suffer from Alzheimer's disease or dementia and which medicine is there? I mean, is there a common rule? Does one lead to the other, or are there common biology that kind of combines these two?
Yeah, just speaking on behalf of what I have learned and patients I have seen, Alzheimer's disease is not like a one-size-fits-all umbrella. Normally when you have Alzheimer's disease, especially advanced Alzheimer's disease, you also have a lot of other issues. So there's definitely been cases, I feel like, where it's Alzheimer's disease and then you also have a tumor, or you have cholesterol issues or heart disease or other dementias as well. I wouldn't say you only have Alzheimer's disease. Normally you see a plethora of different diseases with it.
In your freshman year, did you first get involved in research through an UTRA or in some other way?
No, so I had an UTRA my junior year because I did an independent study through the Butler Hospital. So my freshman year, I just started cold emailing different labs, and I found the memory and aging program at Butler to particularly interest me, and I got lucky enough to be able to interview with them. And that was the right fit for me. So there's a Vivo at Brown website. So it's like just researchers at Brown. If you Google that, it'll come up with a list of every single researcher at Brown. And it has their emails and publications and abstracts of their works. So you have the ability to filter through different things that you want to research. So I knew I wanted to do Alzheimer's disease research, and I filtered through that and just started emailing people. And that's how I got it. Yeah. Yes.
Would you recommend research for a student that isn't a neuroscience major, isn't necessarily going to do this as a career? Do you still see people pursuing research?
Yeah, definitely. I have friends that study English, history, and still do research in that field. Research is very all-encompassing. If someone is interested in a specific topic, I definitely see how it can be rewarding. Any other questions? Cool. So I'm going to pass it over to Hannah. Thank you guys so much.
Hello, everyone. Can you all hear me okay? So my name is Hannah. I graduated from Brown in 2022 with my SCB in cognitive neuroscience. I'm currently working at the memory and aging program at Butler Hospital. I am the senior research assistant on the BioFinder Brown study. So I'm just gonna chat with all of you about how I got involved in research at Brown as an undergrad, my work at Brown as a researcher, and then what I'm currently doing now. So I was really interested in research before I even really knew what I wanted to research in or what my concentration was going to be in. I saw a lot of my peers getting involved in research. Natalie discussed how she got involved in research very early. I found myself very drawn to research methods type classes. One type of class I should say that I would highly, highly recommend to anyone interested in research is the CURE classes. So back when I was an undergrad, I think there were only a few of them, but the program has really, really expanded since then. They're basically classes, they're lab classes, but not the traditional cookbook type lab class where you're running experiments where you already kind of know what the result is supposed to be. CURE classes give you the opportunity to review the literature in a specific topic, develop your own hypothesis, your own methods, and conduct your own experiment and write your paper on your findings. So I found that to be very rewarding. I know now they have them specifically for freshmen, under 1,000 level and over 1,000 level courses. So if you really want to get a taste for what the research process is like, I highly, highly recommend that. By the time I was in my sophomore year, I knew I really wanted to do research, but I wasn't sure how to get involved with it. So I actually took a class called toolbox of scientific research. How do you get involved in research as a half-credit course? Because I just wanted someone to tell me, like, what's the standard operating procedure? How do I get involved in research? And it was a very rewarding class. I enjoyed it very much. I got to talk to a bunch of different people about the work that they were doing, met a bunch of other students who were also interested in pursuing research. And at the end of the day, what the class taught me was exactly what Natalie just told us, which is you need to cold email people. So that is what I did. At that point, I knew that I wanted to pursue a degree in cognitive neuroscience. So I looked at, the CLIPS faculty, but you don't have to be restricted to your specific concentration or department. The advisor, the mentee I eventually ended up working with, was actually in the neuroscience department. So if you have interest in a specific topic, I recommend just casting your net as widely as possible. But I did end up working for Tara White at the White Lab of Effective Neuroscience here at Brown by emailing her. I think I felt a little bit nervous or hesitant to do so, but turns out researchers really, really love talking about their work and that no one will mind. So I got involved with her. I started at the very beginning of the summer between my sophomore and junior year, working on a systematic literature review. So we looked at transcranial magnetic stimulation, or TMS, as a novel treatment for depression. We also use this technique called magnetic resonance spectroscopy, or MRS, which measures levels of neurometabolites in the brain. And so looked at studies specifically that used TMS as treatment and then did MRS before and after to see what are the neurometabolic changes happening in the brain, what are the neurometabolites that are potentially mitigating these depressive symptoms or the alleviation of these depressive symptoms. And that I did my summer between sophomore and junior year, like I said, but it ended up being the first paper I ever got published years down the line. And I continued to work in that lab. One thing that I really enjoyed was getting to do poster presentations. So that's one thing that working in a lab affords you to do. My first ever presentation was actually during COVID over Zoom. So it was the Public Health Research Day. That was the first time I ever presented a poster. Another really great opportunity you have working in a lab is the UTRA, like Natalie discussed. So I did an UTRA award summer between my junior and senior year. And that was really my first experience I had, I guess, technically working as a researcher. You get a stipend, and then you are able to just conduct research the whole summer, work on your original project, which was extremely rewarding. Something else I would really recommend is independent studies. That was a big part of what I enjoyed about being in a lab. I did my first one first semester junior year, and then I continued to do one every single semester until I graduated because I enjoyed them so much, because it just gave me the opportunity to conduct the work and the research that I was interested in, and really dedicate the amount of time to it that I would dedicate to any of my other courses, which was very exciting. A couple people talked about, how being in a lab, in undergrad can give you these opportunities and connections, was absolutely true. I stayed with my lab after graduation for a while before pursuing research assistant opportunities and then eventually starting at the memory and aging program. So I can talk with you guys a bit about the work that I'm doing now. Dr. Huey discussed with you the upcoming exciting research being done on biomarkers. So I am the senior research assistant on the BioFinder Brown study, and we are looking at plasma and retinal biomarkers of Alzheimer's disease, the amyloid and tau proteins. We are especially interested in plasma p-tau 217. I think I've learned a lot working there. I feel like the main takeaways that I've had is that very often you think like where research is going to lead you, and very often you have to change course. So when we started the BioFinder Brown study, we were kind of looking at plasma p-tau in two different boxes, like, oh, this is going to be the threshold, like above this threshold is plasma p-tau positive, and below that threshold is plasma p-tau negative. And that ended up not being that simple at all. We entered this very exploratory stage in the study where we, really redefined what we considered to even be plasma p-tau positive, looking at these people who were cognitively normal and yet had this elevated amyloid, people who seemed to have low plasma p-tau yet came up amyloid positive on PET scans, and doing protocol revisions, IRB resubmissions, and just getting to be involved in every stage of that research process. The other thing that has been really great working at the Memory and Aging program is getting to see people doing all different kinds of work. So we have people, PhDs, the PhD in like neuroscience, cognitive neuroscience. We have neuropsychologists and clinicians and physicians all like doing work together and collaborating together to do this work, which is really exciting, getting to see all these various career paths that research can lead you down. And then of course, the clinical implications of the work. So very often, I always just thought of research in isolation, but working here has really shown me that the next stage and what we do with the research, and we might know that there's a correlation, between plasma p-tau and Alzheimer's risk, but what does that actually mean for people? What are the clinical implications of that? So knowing, if someone has very low plasma p-tau or very high plasma p-tau, maybe that's very clear cut, but for all those people in intermediate range, what do we do with them? Like, what does treatment look like for them? And then the last thing I will also say is, again, just a lot of the people I work with are also at Brown. And I still have a lot of the connections from Brown from when I was an undergrad. I actually talked on the phone 3 days ago with my undergraduate mentor, who I did my senior thesis with, just about taking the research that I did for my senior year, and turning it into a paper. So what I will leave you with is just that the connections that you make here at Brown, you will continue to keep with you over the years, even after you leave and after you graduate. Yeah. Any questions before you all scurry out? Okay. Yeah. Yeah, so I'm currently applying to PhD programs, specifically PhD programs in clinical psychology, which is not something that I realized I wanted to do until I did research. So a lot of people go straight into their PhD programs after undergrad. I really wanted to work for a couple years in research, decide what I wanted to do. And in doing the research and having the participant interactions that I have, I realized how much I enjoy working with patients. And so that made me realize that I wanted to pursue a clinical degree. So I'm currently doing that with the hope of starting my PhD program in the fall of 2024. Yeah.
The challenges in plasma is the proteins, the tau, the difficulty penetrating the blood-brain barrier. So what is happening that the plasma tests are now able to pick that up? Is there a tech change in the plasma? Are they looking at different things, or has there been a technology evolution in how they do plasma? I mean, what has changed, right? One of the reasons why they look at CSF was because of the blood-brain barrier. So what has changed now in the last 5 to 10 years that plasma is able to pick up?
I mean, in your perspective on it, I mean, a lot of it is looking, so the numbers, so it's like p-tau 215, the phosphorylation pattern, and to basically taking sites to look at that are getting higher than you. I don't think I can tell you more than that.
Yeah, that's also pretty much how I know about it more in depth. I know that people have different blood tests, so we're also working with Lund, and we, have to convert our values. And one thing we're actually doing now is our samples are processed by Eli Lilly, and so we're taking samples that were processed at Lund in Sweden and having Eli Lilly analyze them just to make sure we're having concordance. But so, while we know that these blood tests are getting much more accurate, there are still different ones being done. And then I think as time goes on, we'll refine it more and more and see which ones are most effective and most predictive. Anyone else? Okay, well, thank you very much. Thank you, Dr. Huey and Natalie, and thank you all for coming today.